Hello, everyone. Welcome to the NYSCF Internship info session today. We're going to be talking about the nuts and bolts of applying. So we at the New York Stem Cell Foundation are very excited to speak with all of you regarding our annual summer internship and the process that comes with it. Today, we'll have a very brief administrative and programmatic overview of the program. As well as have presenters from our variety of different kinds of internships present, and they will present on what they've done in the past. So all of our presenters are previous mentors, so they will know the best about the projects that interns have led, as well as what they look for in an application and in a candidate. You may have read our slide when you were logging in, but just a little bit of housekeeping. This is a very tight hour, so if you have any questions at any point, you should feel free to place them in the Q&A. We'll also have a dedicated question and answer session in the last 10 minutes. So that's the time where we'll go through all the questions that have come in through the Q&A, and as well as some questions that have already been submitted through the registration process. And thank you so much for those who have done that. And finally, we're recording the webinar today, so you'll be able to find it under past events on our website. The only request outside of that I have from all of you is to ask questions. We would really love to make your application the best it can be. So if you have a question, chances are others do too. So please ask away. And we look forward to seeing many of your applications very soon. So with that, I am just going to start my portion of the presentation. Just one moment. Everyone can see the right view. Okay, so yeah, I forgot to introduce myself. My name is Richard. I'm the Director of External Programs here at the New York Stem Cell Foundation. I'm excited to go over the program and a little bit of the run of show today.
Hey, Richard, do you mind clicking swap displays?
Oh, sorry. I'm totally don't know how that happened. Does that work?
Looks great.
Okay. All right. I am so sorry. Does that work?
Yes, looks good.
It's advancing. Okay. So just wanted to give everyone the presenters that are joining us today. The run of show is that for the first 10 minutes, we'll have an internship administrative presentation and program details from myself. Next will be Valentina, who'll be presenting on the laboratory research track. Corvus will be presenting on the programs administration track. Jordan will be presenting on the engineering track. And Bianca will be presenting on the data science track. So these presentations will be about 10 minutes each, and then the last 10 minutes, again, questions for our mentors and any administrator as well. So again, please ask through the Q&A for those of us joining us, and we will. Okay, so a little bit of a history behind the New York Stem Cell Foundation. In my opinion, it always helps to have the background knowledge on the organization when applying. So this way you don't have to start from scratch. So we achieve our mission through three main programs. The first of which is our innovator community. We externally support early career scientists in both stem cell and neuroscience research. We do this through a variety of award grant-making programs, fundamentally believing in that supporting the best science in the hopes to achieve our mission. So we've built a community of over 200 researchers that are constantly in communication with one another. The second way in which we achieve our mission, or aim to achieve our mission, is the NYSCF Research Institute. So in addition to funding the research externally, we also have our own laboratories with over 100 scientists and engineers. This state-of-the-art facilities houses what we like to call the NYSCF Global Stem Cell Array, which is an automated robotic platform that creates stem cells from a skin or blood cell of a patient, which you can read about online. But just wanted to mention that we also have a number of PIs that are leading efforts in many areas of disease research utilizing these stem cells. And then lastly, something that's near and true to my own personal beliefs is the NYSCF's education and outreach. So we believe that educating all audiences, the public, scientists, students. That's an absolute necessity when furthering the mission. So with that, the one most pertaining to you will be the students. So this is an umbrella of the NYSCF Academy for Science and Society. So today, if you take nothing else away from this presentation, it's this link and this email address. This has our entire internship application, as well as all the information that you need to apply, eligibility criteria, the questions that you have to answer before you even start an application. So short link, nyscf.org/summer internship. If you have any questions at all, myself or a member of the team will be at the other end of internship at nyscf.org. So please feel free to shoot an email that way. So a little bit about the internship itself. It's a 10-week summer enrichment internship program that provides students a glimpse into the fast-paced world of stem cell research. So there's many hats that are of NYSCF. So there's an insight into life at an entrepreneurial nonprofit research institute. There's exposure to career opportunities in science, and also perhaps the most important, connections to valuable professional and peer networks. So for the 2023 program, NYSCF plans to offer primarily on-site internships. So we are still monitoring the pandemic closely, and we'll update selected interns accordingly. But opportunities for virtual or hybrid internships are also possible, dependent on the track, and I'll get into that a little bit later on. It is a paid internship, so we have our payment structure on our website as well, and you can head on to nyscf.org/summer internship for that information. And then just for our internship programmatic tracks here, we have 4 distinct ones, and these are representative of the presenters that we have today. First of which is our laboratory research, where you'll be working within the lab. And because it is a research project oriented internship, it will be an on-site internship. Second, which is the automation or software engineering. You can, if you choose to go down this track, you can choose either one. And in a similar way, automation is very hands-on and will need to be an on-site internship, whereas software can be done virtually, hybrid, or on-site. The data science internship is a hybrid or on-site internship, and this is really working with all the data that's generated from the NYSCF Global Stem Cell Array that I mentioned earlier, which has tremendous amount of data that needs to be analyzed daily. And then lastly is the program's administration track, which is an admin track. This is being done virtually, hybrid, or on-site, and it's really working within the NYSCF headquarters, if you will, really working and understanding how a foundation works in all of our various departments within NYSCF. The program itself, in addition to your project, has there's a extensive curriculum. We will be undergoing a lecture series and a conversation series that virtually every member of the organization participates in. We also have a variety of career development resources. So like a LinkedIn and resume review. We have a crash course on how to present. We have mock interviews. We have mandatory weekly coffee chats with a variety of NYSCF staff. So that all culminates at the end, the last week, where we have a presentation day where interns will present posters like you see in the picture today. So application-wise, this is essentially what it looks like. We have essay questions that are pertinent to each track. So each track has different questions. Please look out for that. We also have a cover letter, resume, transcript. It can be an unofficial transcript. And we ask for at least 1 letter of reference, but you have the option to upload 2. And all applications must be submitted through our portal, which you can find on our website at nyscf.org/summer-internship. I'm not going to stop saying it. If you find our opportunity on Handshake, please do not apply through that portal because we won't see it. So if you see it on Handshake, then just go over, head over to our website where you can go through the portal And the deadline for the 2023 cycle will be January 6th, 2023, which is a Friday. This is just what it looks like when on the portal itself. So if you see this page, you're heading towards the right direction. Just wanted to point that out. And you can also preview our application without having to create an account. You can actually find the link right down here when you log on to the NYSCF portal. And we have a timeline here. So applications opened at the beginning of the month. Today is the 15th, which is our information session. Applications and letters of reference are due at the same date, January 6, 2023. We are really hoping to notify all candidates by late February. So that's our goal. And then the internship will start June 5th, 2023. I'll go on for 10 weeks and end August 11th. With that, to Valentina to speak about the laboratory research track.
Sure, thanks, Richard. Let me share the screen. You guys see my screen, right?
Yes.
Okay, everyone, thanks. So I'm Valentina Fossati. I've been at NYSCF for over a decade, and I work with my team on neurodegenerative diseases. I think that all of us, unfortunately, are familiar with neurodegenerative diseases. Either we have somebody in the family or we know someone that is affected by one of the common neurodegenerative diseases, thinking about Alzheimer's, Parkinson, multiple sclerosis. Those are devastating diseases for which we don't have a cure yet, and they are affecting several millions of people just in the United States. And so the way that at NYSCF we are looking to understand and to cure these diseases is using the power of stem cells for making human models. So we are able to recapitulate and study the disease in a dish. And I think, probably many of you or all of you know that over the last decade, since 2007, we have been able to make stem cells in a dish, just starting from a few drops of blood from any of us that can donate. And so once we make those stem cells, we also are able to generate all the different cells of the body that we want to study. And so, as I said, because I'm working on neurodegenerative diseases, I have been interested in making all the cells of the brain. And you are probably familiar with the neurons, which are our communicator cells that are allowing us to think, to move, to do everything we do. But the brain is actually a very complex organ that has many other cell types inside. You may or not be familiar with the other cells. Main cells are like oligodendrocytes, astrocytes, and microglia. And we work every day with all these cell types. And if you're coming for the internship, you're going to know much better about them. And so we first learned the recipes to make those cells in the dish. And now we are working on seeing how these cells function. And we are working on creating those miniaturized parts of the brain, which we call organoids, in which we put all the cells together. And so the interns that come and join our team usually work closely related to the scientists that are with me in the team. And they have a specific sub-project, so a little part of a much broader project that is able to complete within the weeks of the length of the internship. And they perform their own experiments. At the end, they have to collect all the data and they learn how to present this data. As Richard also said, we have these poster sessions where all your data are summarized together. And this is, for example, the poster that one of the interns, Anna, made in 2019. And she was working again on these organoids, trying to recapitulate the cortex of our brains. The day of the poster presentation, there is also an official talk. It's called poster teaser. It's a 3-minute talk in which you have to really quickly summarize what are the claims, what you discovered or what you worked. And again, this is another example of the teaser from another intern from 2021. Again, look at the beautiful images that she was able to take, and so she learned a lot about confocal images and about how we can study and characterize all the different cells in the brain. And I have to say that I particularly love this internship program because a lot of times the candidates that are applying for the internship are actually interested in coming back and working at NYSCF. And this is the case for especially for people that wants to continue their education with PhD or MD PhD programs, and so they want to do a 2-year gap to really acquire experience in research. And I had 3 of these wonderful summer interns that came with me over the years, Madura, Lily, and Tanya, that are now rising stars, really. They all entered in the program in the school that was their dream. And they are striving there, and they are almost even so at the Tanya is almost at the end of her PhD, and so we have kept the communication with them, and they all, I have to say that all these interns, they become very good friends, and they are all, many of them are still in touch after many years. So I think the questions that the people that are listening here today is like, how do we select? Because this is a very competitive program. We're going to receive a lot of applications. So how do we pick the persons that we want? For me personally, there is only one major thing, that is showing your passion. I really try to look for people that are interested in working with stem cells, in working with a particular neurodegenerative disease, in people that really wants to come and do this experience, not just that can be, any of the possible internships. So you can see sometimes you really see the fire in the eye of people, and that's what I always try to find. Obviously, there are different. My slides are not. Okay. I really much look forward to read the cover letters. Please pay attention in making your cover letter and really make it personal and explain why you want to come to this internship. And I know that you're applying probably to many, and it's tempting and it's easy to just make one letter and then copy and paste. But we can see that, and actually sometimes I read some letter that says, "Dear Dr. Smith," we don't, at least remember to change the name and to address it to the right person. But what really makes a difference is reading through the lines. I really try to see through the lines why you want to apply to this lab, why you are interested in coming, particularly working with our team. It's definitely nice. It's a plus if there is already some general knowledge on stem cell biology, previous experience with cellular molecular biology, but it's a plus. It's not really a requirement, and that will change the type of experiments that you can do. Obviously, if you already have some experience, your training will be shorter, and so you may do some more experiments independently. So that really changes, may change a little bit your actual project. And then, science in general, but in particular our work in my team, it's really a teamwork. There are several people working in projects that are very much overlapping. So we need a friendly attitude. We need somebody that works well in a team, cooperates, and are able to work together. And really genuine curiosity. I love to see enthusiasm and people that is willing to learn and yes, and embrace this experience, which, talking to a lot of interns after, they always say this is one of the best period of their life. They really, really enjoyed it. And so we are very excited to have you here with us. And I'll stop here.
Thank you so much, Valentina. So if you have any questions, Valentina, and pertaining to the laboratory research track, again, we have the Q&A feature open. Next, I'm going to pass it on to Corvus, who will speak to you about the program's administration track.
Hello, everyone. So my name is Corvus Richardson, and I've been with the foundation for 4 years now, and I oversee the foundation's intellectual property and other legal affairs. And so when I say intellectual property, what I'm talking about are patents, copyrights, trademarks, trade secrets, more generally know-how. And these are all the things that are being generated from the great work that's happening within the lab. So some of the things that Valentina even mentioned in regards to the recipes for our ability to make different cell types on our automated system, the NYSCF Global Stem Cell Array. And also in regards to our engineers and what they're doing, data scientists, and really trying to leverage the value that's generated. And so one of the motivations for the foundation being interested in protecting intellectual property is so that we can move these technologies towards the clinic or for the benefit of patients. And so that's our in line with our mission and our objective. And so that is one aspect of what I do. And then in regards to another aspect, it's more general in regards to legal matters, so contracts, review for compliance, and other things as well. And with that, I work very closely with our business development, which will have their own intern in regards to that, and as well as with our finance team, our grants team, development, and the like. So working to put in place the necessary agreements for certain collaborations. We also we have the ability to make induced pluripotent stem cells, iPSCs, and are able to make that available for research purposes. So in doing so, we enter into what's called a material transfer agreement. So I'm involved in that process as well. And this will be coming up, would be, I believe, my third summer of interns. And with that, it's actually been a really great experience. And I like to say that those who are able to join in the summer and work with various groups within the foundation, you'll be working on real projects that will continue on even after you. And so it's not like you're given busy work. It's really impactful work and active work. So a few projects that we've worked on over the summers, and some of them are actually still ongoing and also expanding. But one summer we were working on an investigator database program, or creation of a database for tracking. And Richard and his team are heavily involved in this, but we provide funding for early career scientists. And with that, we enter into grant agreements. And with that, there are certain terms and just general obligations that the investigator has to meet in regards, and also for us to track. And so we created a database for us to be able to track IP that's generated from their efforts, as well as their publications and the like. Also, this most recent summer, it was focused on IP tracking and analysis, and it was a very impactful project just because now we're moving into the phase where we can model budgets and kind of look historically to see where our spend was on in regards to the different technologies, and we can project forward in regards to what we expect to spend. And this also helps drive strategic conversations related to where we can and should allocate resources in regards to IP protection. Another component that has been really helpful is legal research and compliance. And so I've had interns help with this effort in regards to performing a bit of legal research on certain topics that are essential for what we do. And so there's a program that we're working on, and the New York Clinical Laboratory Evaluation Program, or CLEP, is one where we were evaluating what the requirements were for New York State in regards to being able to register as a clinical laboratory, and understanding the specific requirements there, in addition to the New York State Department of Health and Tissue Bank program, and being compliant there. And so the interns they were able to basically draft a legal memo, and I helped them with that in regards to understanding what the components that go into that, and that is also something that can be shared internally with the necessary stakeholders. Another area that we've worked on has been like market research and analysis, and this has actually been in close collaboration with the business development team. The business development and IP legal work really closely together. And so one particular summer, we were looking at cryopreservation, so the ability to freeze cells and thaw cells. And we were specifically looking at just expanding NYSCF's ability to cryopreserve different cell types, so cell types that are generated from the iPSC themselves, so neurons or microglia or astrocytes, similar to what Valentina was saying, and that potentially could expand our ability to make those available for research purposes. Another project that we worked on was in regards to looking at our patent portfolio and the technology, and understanding the landscape to see what other people are doing within the space. That's important, I think, from a technical perspective in regards to what the researchers are doing, but also in regards to patentability and the patent landscape, and also business development for potential collaborations or understanding potential deal structures and the like. So often each summer, my intern actually collaborates on a project with the business development intern, and that has proven to be really impactful and beneficial. And I believe that the interns enjoy the opportunity to collaborate on a specific project, and to kind of see how our team really collaborates and works together. And these are just a few that I've highlighted that I think are pretty interesting, and also gives you a better view as to the different things that can kind of go into the legal IP track. It's not just purely legal or purely IP. There's other considerations and other tasks that you might be able to work on, and it's all current and relevant topics to kind of engage in. And in regards to what I look for in an applicant, I think your interest generally in law, your interest in intellectual property is something that I do look for. I've worked with individuals who have very little experience with IP, and others have more experience. I think it really just depends on you articulating what in fact you hope to get out of the internship, and also the current programs and projects that we're working on. Those are a few considerations that go into making a decision. And I would just encourage that if you're interested and just unsure, go ahead and apply. I would even encourage you to relay that within your application, the thought process behind you applying for the legal track. I think that would be very interesting to learn more, and also to learn more about you. So I do encourage everyone to apply who's interested, and we'll hand it off to Felipe Bianca or to Jordan.
Thanks, Corvus. Yes, as Corvus mentioned, if you have any interest in legal or intellectual property, please do apply. For the programs administration track, in addition to the legal and IP, as Corvus mentioned, we have usually opportunities in the business development team. We have scientific communications and outreach, nonprofit program management, fundraising. They're all on the website. So if you're interested in any of those, please apply. Next, I'm going to hand it off to Jordan, who will be speaking a bit about the engineering track.
All right, thank you, Richard. All right, let's see here. 1 second. Okay, welcome everybody. My name is Jordan. I have been here for 7 years now. And I actually started off with a biology background, but now I work along with a really talented group of engineers with a really varied expertise in a lot of different fields. And that really allows us to cover a lot of ground. So I'll talk about a little bit of the different things that we work on and highlight some of the projects that interns have helped us with over the last year, a couple of years actually. So just to give you a bit of background on what we work on here, my group kind of works within this large ecosystem of a lot of different teams. So you can see that this number is maybe 30 to 40 people, all working very closely together. People work with computer science as software developers, data science teams, as you'll hear from Bianca in a little bit. We have a lot of hardware engineers, sort of process developments, process engineers and programmers. And really, we all work alongside the biologists who operate the equipment and do the screens, differentiations, and complex experiments that use the platform that I'm about to talk about. So all of these groups sort of work with and around this kind of system that we like to call the NYSCF Global Stem Cell Array. So as you can see in the video, this is really a collection of really advanced robotics that really handle a lot of the same work that biologists normally do by hand. But in this case, we can have a lot of different equipment working together to really scale up and make a lot of these processes very defined. In addition, a lot of this work can be done sort of remotely so that we can generate a lot of data points and metadata and images. And so it's a very powerful system that I'll explain a little bit more about in a second here. But really, as you can see in the video, it's a lot of equipment like liquid handlers, robotic arms, centrifuges, incubators, imagers, and all those kind of things. So taking a step back, what this really allows us to do is really collect material from a lot of different patients from different demographics and gender. And we can take either their skin or blood cells and use all that equipment that you just saw in motion to really make a lot of different cell types that represent the disease of interest and kind of run a lot of experiments with those cells. And what we really like to focus on here is the population scale. So you can see that whereas some labs like to really scale up, one patient line at a time, we like to survey a wide variety of different patients. So just to give an example of that, if you consider one patient in this single flask here, that you can maybe do 10 at a time if you're really good, but we like to extrapolate that into, a full 96 well plates. So imagine 96 different patients in that one plate. And we can even get up to 384 samples, or taking that even further, sometimes we have 1,536. And, if you think about all those plates in the picture there, using our system, we have maybe a couple hundred plates in culture at a single time. So you can really see the principle that we try to go for in our research is really large scale experiments here. So, how do we go about maintaining this equipment and trying to improve it? We really focus on making, expanding the functionality of all the equipment, and making it more reliable and easier to use. So, we focus on a lot of programming work for a lot of our liquid handlers to make these protocols and to allow the different instruments to talk to each other. And this is where some of the intern projects might come into play. We have a lot of those machines that you have their own sort of proprietary integrated development environments. But we also have some liquid handlers that are very open source and easy to use, which I'll get to in a second here. So for example, some of them can be programmed by anyone who's had exposure to Python, let's say. We also like to have people help us make custom packages and tools that really make the use of the equipment easier. So, for example, parsing trace files or data logs and stuff, making work lists and graphical user interfaces, and really adding to functionality in different driver software that we have to allow more flexibility and modularity. Alongside that, we also work on projects that involve 3D printing and really some hardware fabrication and maybe microcontroller devices. So I'll get to some examples in a second here, but we really kind of we wear a lot of hats here, and we're able to work on a lot of different things. So like I just mentioned, we do have a couple liquid handlers that are really easy to put together these experiments with. So one example on the left is this Opentron system. It's a really kind of benchtop, affordable system that you can do a lot of advanced experimentation on. And this takes Python and JSON programming. So we've had some interns in the past build protocols using custom labware and really fine-tuning all the liquid class parameters for just to make different reagents work together. So that's been one example is this is the kind of programming environment that they use. So it's very user-friendly. We've also had some interns that have helped us with sort of verifying the volume, that the pipetting performance of these instruments, just to make sure that we're getting the right materials and volumes of components in there. So this is an example of using some fluorescent dye with a reader that we can set up these QC methods to test the volume with, and that really ensures the quality of our pipetting. We also have there are modules that we can introduce that will help everything run smoother. So an example on the left there is another liquid handler that we use you can see the tips, the spent tips are just getting built up and accumulating. So we've had a couple interns that have sort of built a tip chute that kind of ushers the tips down into a garbage can externally. So, you know, an example, this is work from an intern from 2020. We measured the design of the waste bin and figure out what kind of assembly to use for this to solve this issue. So an example here is you know, a sloped chute, and you can see right on the front there is a ultrasonic proximity sensor that detects when tips are dropping down that collar, and there's a vibrating disc motor on the chute that kind of ushers the tips down the chute. So we've had interns kind of learn to use SolidWorks, which is a CAD software. So you can see this is kind of just designing the different components, and then we move them onto a 3D printer that we have in-house that we can you know, put together these different components. So the finished product you can see right there is it fits very nicely, and it has all the components that you need. And I'm going to skip the video here, but that's another view from the front of the system there. So another example, making devices with microcontrollers is this tip vacuum waste line solenoid valve. So this is another example of a liquid handler that we have that we use very extensively, and the waste trough that is being labeled in the picture is where a lot of the media goes, and a lot of the reagents and washes go down a plumbed vacuum line, and it collects on the left side in that waste reservoir at the bottom that's pre-filled with a little bit of bleach for neutralization. But oftentimes they might overflow, or the user forgets to switch on the vacuum line. So to make everything really streamlined, we've had some interns design an automated waste valve. So we can either, we have the actual liquid handler system actuate this solenoid valve that will turn on the vacuum flow by itself. So we've had the interns play around with these Arduino boards and Raspberry Pi boards, and really just iterate over a lot of designs. So we integrate all of that with the 3D printing kind of enclosure, and you can see some of the Arduino code there. It's very easy to play around with, and a lot of fun. And then just one more project that I'll point out. Another thing to do with some of our waste material. We have these bins that collect all the tips. And as you can see, this is at the bottom of some of our biosafety enclosures. And with the door closed, it's kind of hard to tell when the tips are overflowing the bin. So an intern this last summer put together this a scale that will constantly measure the weight of the bin, and after it hits that certain saturation point, it'll flag the user to empty out. So a lot of devices that helps make everything just work better. So we also have a software development track alongside the automation engineering track, and so this this has to do with our in-house software toolkit that our team has developed here, and we call it, it's a web-based application that interacts with a lot of data tables and databases to communicate with and kind of orchestrate a lot of operations on the equipment. So examples of some of the projects that are being developed currently are these calendar tools that help really organize a lot of the runs for that day. You can see all the different systems that we have. There's a lot of work that has to be planned out, a lot of experimentation. So this is just a way to organize everything. Additionally, there are these work list generators that are very graphical to allow people to track and plan out where their cells will go in these complex protocols. So a lot of these tools are built using React and .NET Core as the base, but then there's a lot of hooks and integrations that use a lot of different languages like C#, SQL, React, and TypeScript. So those are just a couple of examples of some of the software programs that we have. So, just to point out what we look for in certain applications, of course, we like to see some relevant exposure to the languages that were just previously mentioned, but not necessarily expertise is required because we want you to learn a lot on the job. But I think more importantly, maybe is the demonstration of communication skills. So the array in general is very team oriented. So you're required to work very closely with all of those different people that we mentioned before. So really everything is going to be impactful, and you need to understand what the user requirements from every different group. But also we just really like to see people who are motivated and like to take on this work in their free time and outside of normal coursework, if you have any hobbies or side projects that you want to highlight. So those are some of the things that we look for in the applications. So yeah, be happy to answer any questions during the Q&A period.
Thanks so much, Jordan, for those examples. A lot of projects from the past. Okay, and then last but not least, I'm going to hand over to Bianca, who will be speaking on the data science track.
Okay, can you see my screen? Okay, so hi everyone. I'm Bianca Migliori, and I'm going to talk about the data science track. As a first thing, I want to highlight that this is not just like coding or just software engineering. It's more like trying to use the data and use computational tools applied to it to uncover something new within, of course, our scope of research. So first of all, I mean, Jordan spoke extensively about our automated pipeline. So this is an example of how one of these pipelines look like. So we have our patient. From our patient, we derive some cells that we can derive into virtually any cell. And then we can do some experiments over them, which would be like cell replacement therapy oriented or drug discovery or disease research in general. What is important for data science is the scale where like Jordan and the team is working on. We need a lot of data to uncover very specific phenotypes of diseases we're looking at. But in order to have that, we have to kind of make sure that everything along the pipeline works correctly. So we can work as a data science team into different levels. The first level is within the automated pipeline. So for instance, as the cells grow, we can image them every night, or we can like do something to them every night, every day, or in an automated fashion. So our robots go take the cells, take pictures, and analyze some factors. And what we can do is to implement some machine learning algorithm or some algorithms in general to make sure that all of these images were taken correctly, or the amount of cells were right, and there are not too many cells and so on and so forth. So this is the first aspect we can work on. Then there's, of course, another step of quality control. So is the result fine? Is the outcome of the experiment the desired one? Can we use these cells to further analyze them? So there's a lot of things we can do here to make sure that everything went correctly. The density was fine, the colors were applied correctly when we want to look at stained cells, and so on and so forth. And then last but not least, we can uncover new things from this data. So in the end, we have this massive data set that would be really, really hard to analyze by eye or by hand. So we need to design some algorithms or some computational tools to make sure that we are able to get the information we are looking for in these data sets. And this can be different, like it can be something where like you have a hint that maybe you want to measure some specific organelle within this cell of interest, or maybe you want to understand if there are any differences between like some complex diseases. So that's what we work on in our team. We work with many teams and with very different cell types. So first of all, one of the biggest area is in fibroblasts, which are just like cells that they take from the skin in a very or low invasive way, so that we can look at them from, so they're primary fibroblasts or adult cells, and they also express all the epigenetics that each person comes to at that specific time. So we looked at Parkinson's disease in these fibroblasts. We are also looking at INA disease, which is an infantile neuroaxonal degeneration disease, and aging, and among others. Then we also looked at osteoblasts, for instance, to try to work on bone replicates. We worked with ovarian cancer cells. We worked with diabetes cells. And then we worked on neurons, both for like Parkinson's and INAD as before, as well as new projects. So as you can see here, we are already looking at very different cells. So the approaches for the analysis can vary a lot. It can need tweaking in order to work correctly. So just a quick overview of the project that we worked on in the past with the interns. So I joined NYSCF 3 years ago. So I had 3 rounds of interns. This one was from the first year, and it was Jomari Lanzi that was working on an RCNN, so a region-based CNN convolutional neural network, to try and identify different clusters of cells within ovarian cancer colony. So basically, in this case, we did not have any ground truth, which means that we did not know which was what, but we wanted to have an unbiased algorithm to tell us which ones were different, so that we could identify different clusters of cells based on morphology only. Another one from the same year was Paulo Serodio, which did also an amazing job trying to trace neurons, which might seem like not that difficult task, but in reality, the images are not that clean. So there are like speckles everywhere, and there are neurites going all over each other. They can become very, very dense. So what he did was to design this way of like overlapping directional masks over each of the traces. So basically just like a convolutional window. And then he was able to trace the neurons, also giving or measuring the directionality of it, since we saw that in some specific diseases, there was the tendency of organizing towards a specific line. And then skeletonizing, so getting to the core of each of these lines, he was able to trace these neurons in a very precise manner. Then we got Jeff Winchell, which, by the way, we hired. So he's currently working with us, which was working initially on a calcium imaging analysis tool. And as you can see, this is what the images look like. So there are short movies of cells that are firing depending on how their calcium expression is going. So basically, it's a measure of the activity of the neurons. And as you can see here, the images are really pretty, but also very messy. Like there's a lot going on. So it's not that easy to like distinguish all the neurons and analyze them and like understand if they're spiking or not. Because as you can see, whenever you design a region of interest around them, there are a lot of spikes and a lot of activity going on. So he worked, maybe this is not even the best example I could put here, but he worked on this pipeline that was recognizing the neurons very well and also recognizing the spikes. And now he's also working on how to to like see the relationships between them, and if there is like a driver neuron, and if like there is a specific pattern or differences between diseases. And another, this one was an intern from the last year, and Peter McEntee, and he was working on flow cytometry. So flow cytometry is an incredible tool where you can take a bunch of cells together, stain them for something specific, and then you have them go through this microfluidic device where you can read how much they fluoresce. So if you have a large combination of dyes and different types of cells and different type of stains, you can have a very, very dense data set coming out of it. So this is how the data looks like. Usually you have a reference gene, and then you have a bunch of different things you're screening for. And you can see how the intensity of fluorescence varies depending on this reference gene. And then you can do all sorts of things. So for instance, what he did here was to do a 3D clustering using PCA analysis, so principal component analysis, to see if there were specific clusters, to see like how many of these new dyes were similar to the old one, where they were differentiating, where they were similar, and so on and so forth. And here, like other tools to see the distribution of the data set to try and uncover which one of these dyes could be more specific compared to the reference dye. So in conclusion, in a highly automated research lab, there is a strong need for AI, because there are so many, the data sets are so dense, there is so much information everywhere, and there are so many things that can be automated so that we can use less people and also the less variation due to just people intervention. And possible projects in the future can include like AI methods for automated imaging QC and cell counting, but also, so QC is quality control, sorry. Analysis of imaging data of any kind, trying to like read images so that you can get the different phenotypes of the conditions you're looking at. And then analyze high throughput data of any kind, which could be images, could be flow cytometry, could be any other readout that we have throughout our pipelines. We can do any sort of analysis or build any sort of tool that can go and contribute with high content imaging analysis. So for instance, we have like another intern that built a tool that is now integrated in all of our pipelines. So we're grateful for him. Which is Jeff Winchell, which we hired. And then there are also a lot of data organization, cleaning and formatting that are needed in all this process. So like a good candidate for me would need to have like show a lot of passion, as Valentina also highlighted. I also like curiosity. So if someone is very curious about not only AI, but also like what's the outcome of AI, I think that's crucial for machine learning and data science, and someone that is willing to try out things. And I think I'm over time, so I'll pass the word to Richard, and happy to take questions.
Thank you so much. So yes, we're at the 5 o'clock mark, but we're going to go a little bit over. Those of you who have to leave, this is a recorded session, so we'll be sending this out in a follow-up email if you can't stay. It's also available on the NYSCF YouTube channel. Before I pose a question to the panelists, I just see two main sort of administrative questions coming in. If you want to apply to more than one track, we allow applications for up to 2 tracks. You'll have to indicate a priority one and then a secondary one, and then you'll answer the specific essay questions for both programs that you're interested in. So answering that. And then secondly, there are certain individuals asking about, transcripts or not as much research experience or not as high of a GPA. I will say that just programmatically for our internship review process, we take a very holistic approach when reviewing applications. We're not looking at just the resume and just your transcript. If anything, we're looking more closely at the cover letter that you provide that many of our presenters today have mentioned, as well as the answers to your essay questions. And this is why we pose those questions, so that we can sort of gauge into your mind and then see what you're thinking about and what you're interested in. So I would highly encourage you to still apply, and also pay special attention to those sections. So just overall to our presenters in the last 5 minutes, I think I'd like to just ask generally, what would you like to see an intern achieve in the 10 weeks of the program that they join us for? And you can go in any order that you'd like.
I guess I can start. I think for me, I like them to have fun and enjoy, learning some of the things that maybe they're not the strongest at to begin with. I think a lot of the projects are going to be very impactful, and they're going to help us out no matter what. So I think that I'd like to see, really everyone have a good time.
I guess I would say I really enjoy hearing feedback that that they've learned something, that it actually has maybe impacted their direction in life in regards to a career path and stuff like that. Because as I mentioned before, I've had interns who have had experience with IP versus very little to or even very little with like legal to more, and kind of just seeing like that they actually got something out of the internship. I think that's probably the biggest thing that I hope to see at the end of it, that it actually meant something and will further elevate them in their career moving forward.
Yeah, I agree. Me too. I like to know that this internship was helpful and that they learned something, and that they go home with more passion about what they're doing and more clarity about what their future should be. And yeah, I really like also having a fresh eye on things, because sometimes you work on something, you create your own ideas, and you don't always get out of what you see. But with interns, and especially the ones that are curious, that ask questions and that are engaged, many times you have this fresh view and something new comes up, new ideas and new unexpected outcomes, which I really like of the program.
Yeah, I think I can echo everyone. We are primarily here to mentor you and to give you the chance of really being in a state-of-the-art lab. And I don't personally don't have expectations but most of the time, actually, the wonderful part is that most of the interns really do amazing work. And so anything that we come back, sometimes we had people that really did critical work that went in papers, very important experiments that changed the directions of this project that we were going. But we start by teaching you, and then it's a constant feedback and really enjoying and capturing the sense of what is being a scientist or a different role, an engineer, data, whatever, like having for you this to enjoy this experience, and then everything comes spontaneously. I think if you enjoy it, then you're gonna produce a lot. And we are always amazed by the posters and the quality of the work of the interns. So I have no doubts that everybody will do a great job.
All right. Thank you so much to our presenters today. Thank you to all of today's session. And I just want to plug that there is a survey included in that email. Please fill that out. It really helps us to make our events better and better. So thank you all again so much, and have a great night.